Good afternoon and good morning, everyone. I am Tiffany Penn from GenScript, and I would like to introduce our next speaker, Dr. Samantha Yost. Dr. Yost is currently a senior scientist in gene transfer technology, a research and early development department at REGENXBIO. Dr. Yost received her PhD from Rutgers University in 2016 in microbiology and molecular genetic research, focused on structure function studies of alpha-viral non-structural polyproteins. In 2022, she established a biodistribution core group to streamline animal tissue processing and analysis of vector genome biodistribution at REGENXBIO. So for today's topic, that will be characterization and biodistribution of REGENXBIO NAV platform capsids on their employed gene therapy vector AAV7. If there's any questions regarding this presentation, feel free to put them in the chat box at any time, and Dr. Yost will answer them after her presentation. Let's welcome Dr. Samantha Yost.
Good afternoon, everyone. My name is Samantha Yost. I'm a senior scientist at REGENXBIO in the Protein and Vector Engineering Group in Gene Transfer Technology. Today, I'm going to be telling you a little story about a somewhat ignored, underutilized capsid called AAV7 and the establishment of a battery of experiments to thoroughly characterize newly described capsids. First of all, I'd like to thank the organizers for inviting me to speak and thank everyone for watching today. I joined the REGENXBIO R&D team in 2018. I was the first member of the capsid engineering group. At that time, we were planning how we wanted our group to function, what areas of research we wanted to focus on in terms of capsid engineering. So ultimately, we decided on three main focuses, first being structure-based rational capsid engineering, directed evolution of capsids, and lastly, characterization of naturally mined capsids. I believe last year, if any of you watched that, my colleague Ye Lu presented on all of that and our strategies toward capsid engineering. If you did not watch that, I'm sure he would be very happy to send you the slides if you contact me on LinkedIn. But I'm going to talk about mainly characterization of naturally mined capsid, of which REGENXBIO owns over 100. We selected AAV7 for this evaluation as a candidate. We used this as a model to establish a workflow with which we could thoroughly characterize other new capsids. At the right of the slide is a tree showing the relationship between many of the REGENXBIO-owned capsids. The orange arrows show common benchmarks used in the literature, such as AAV9, AAV8, RH10, and AAV7. AAV7, as I said, is a REGENXBIO-owned capsid with very little known about it. Its protein and or glycan binding partners are completely unknown, and previous glycan binding screens in the literature are reported to be failed to generate significant hits. Previous studies that include AAV7 generally have a small focus, are not well controlled, are not compared to well-described benchmarks, contain comparisons with different promoters for different capsids, and in my opinion are just not complete. So by initiating this work, we wanted to inform future engineering studies using structural biology and in vitro screening methods, compare AAV7 to well-known benchmarks, in this case AAV9 in vivo, and establish baseline information to enable the use of AAV7 in a clinical setting in the future. Here's a brief summary of everything I'm going to talk about and some things I'm not going to talk about. Overall, this is the layout of the in vitro and in vivo experiments that we put together for this evaluation. First, we looked at the manufacturability of AAV7 compared to AAV8 and 9, the two capsids that REGENXBIO has the most experience with. I'm not going to talk about this data. Honestly, it's a little boring. It's just a graph. We found that AAV7 productivity was comparable to these two benchmark capsids. Next, we evaluated the potential of potency assay development through the transduction of HEK293 cells overexpressing AAV receptor AAVR. I am going to talk about that on subsequent slides. Unfortunately, the next experiment, we did a 300 glycan array assay. This failed to produce any meaningful hits because of indiscriminate glycan binding profiles for AV7. However, our positive control AV9 did come up with N-terminal galactose as intended. We additionally determined the structure of AV7 capsid via cryo-electron microscopy. As for the in vivo experiments, we sought to thoroughly characterize biodistribution in a mouse model after IV administration, which, despite being a very common assessment in the field of gene therapy, typically using PCR-based methods, we exposed some deficiencies in this usual technique, which I'm going to touch on further in the coming slides. But the spoiler I'm going to give you right up front is that based on this work, we conclude that imaging studies should almost always complement traditional PCR-based biodistribution studies to create a clearer picture of transduction capabilities and tropism for any newly described vector. The potential of potency assay development was evaluated through AV7 transduction of HEK293 cells overexpressing AV receptor. There's some conflicting information in the literature about the relationship of AV7 and AVR, so we compared transduction efficiency to AV8 and AV9, which are AVR-dependent vectors. AVR overexpression increased transduction efficiency of all three of these vectors. For AV8 and 9, about 10-fold. For AV7, a modest 2.8-fold. We hypothesized that the AV7 and AVR interaction may be weak, as further membrane protein binding assays did not identify AVR as a potential AV7 binder, although it was identified against AV8 in the same assay. The AV7 structure suggests, I'm going to talk about that further as well, but it suggests that residue T273 may impact the predicted AVR binding surface compared to AV9. Overall, we conclude that HEK293 AVR cells would support development of a potency assay for AV7. The AV7 capsid structure, shown in blue here on the right, was determined via cryo-EM at 2.7 angstroms. This was deposited in the PDB under PDB ID 7JOT. It was the first AV7 structure that was deposited in the PDB. The structure contains a common fold across all described AAV VP3s that have been deposited in the protein data bank. AAV RH10, shown in red, is the closest capsid in terms of sequence identity whose structure has been deposited in the PDB. The amino acid backbones are largely identical between the two capsids, with one notable difference. Variable region 1, or VR1, a loop previously implicated in conferring the ability to cross the blood-brain barrier, or BBB, highlighted at the right in the orange box. Known non-BBB crossing AV8, shown here in green, has a perfect overlay and amino acid identity of the VR1 loop with RH10, a known BBB crossing capsid. AV7, however, adopts a completely different conformation than both of these two. BBB crossing ability is highly desired in gene therapy, as delivery directly to the brain can be quite invasive, and a systemic approach with additional brain transduction would be highly desirable for many indications. The structure here gives us more questions than answers, honestly, as the VR1 loop adopts a completely different conformation than both of these. Considering some other evidence, such as presence of a conserved serine at position 268 in AV7, shared by BBB crossing capsids, we were eager to answer the question of whether AV7 crosses the BBB or not. So we come to our first in vivo study here. This is a mouse study where we compared AV7 and AV9 via IV administration at a dose of 1E13 GC per kg. Biodistribution was evaluated 3 weeks post-dosing via qPCR. Overall, biodistribution was honestly quite similar between the two and suggested that AV7 can transduce the brain at levels equivalent to AV9. We saw a potential increase in the transduction of skeletal muscle for AV9 via DNA genome copies, which actually evened out on the mRNA transcript level. After this experiment, we were honestly a little bit disappointed that the two capsids looked so similar, but we decided to seek out another approach through direct visualization of protein expression from AAV transduction in a whole mouse model. Cryofluorescence tomography, or CFT, was used to reconstruct 3D models of animals transduced with our vectors conferring expression of fluorescent proteins. It was also used to quantify mean fluorescence of defined organs, as shown here. We analyzed AV7 and AV9, both conferring GFP expression in single vector groups, and chose to dose higher at a 2E14 GC per kg to ensure direct fluorescence visualization would be possible. At this higher dose, and using this method, we saw the persistence of skeletal muscle preference for AV9 over AV7, similar to the previous DD study, as well as increased heart transduction by AV7, and a surprising amount of expression in the mandible region, something you really don't see sampled by traditional PCR-based biodistribution methods. Here, we show the reconstructed 3D models of a representative animal of each group. As I mentioned, we have AV7 and AV9 conferring GFP expression dosed as single vectors. So that would be the first and second mouse. But we additionally dosed a third group with a mixture of AV7 GFP and AV9 TD tomato as a pilot to evaluate the CFT method in the context of dual fluorophores. Note that due to the difference in fluorescence intensity between the two fluorophores, this group is not meant to make a direct comparison between AV7 and AV9. However, in the context of the single vector GFP groups, it does aid in visualization of localization of AV7 for heart and AV9 for skeletal muscle. These are the CFT models reconstructed for the heads of each representative animal, which were imaged separately from the bodies to gain higher resolution. As you can see, there's a lot of transduction present in the mandible region, which appears to be teeth, incisors, olfactory organs, or potentially some jaw muscle and tongue. As the CFT technology was quite new to us at the time, we wanted to further validate these results by recapitulating the higher dose study using immunofluorescence and immunohistochemistry. We dosed AV7 and AV9 conferring GFP expression at the higher dose of 2E14 GC per kg and examined heart and skeletal muscle. The study confirmed the superiority of AV7 for heart muscle and AV9 for skeletal muscle for gastrocnemius and tibialis anterior. To investigate further the fluorescence seen in the mandible of the CFT study, we performed immunohistochemistry on sections of decalcified mouse head and found high expression in GFP for both AV7 and AV9 treated animals in the tissue of the olfactory epithelium, seen in B and G, muscle cells of the tongue in C and H, and odontoblasts in the incisor root in D, E, I, and J. As these are areas not typically sampled by PCR-based BD methods, this would not have been explored at all without insight from CFT. So far, on the BBB crossing front, we have conflicting information from the structural and sequence evidence. The lower biodistribution study showed similar BBB crossing abilities of AV7 and known BBB crosser AV9. And the CFT study showed negligible differences between AV7 and AV9 in the brain region at the higher dose. So is AV7 really good at crossing the BBB and transducing the brain? And is it as good as AV9? Well, no. Shown here is GFP expression amplified by IHC in the CNS following IV administration of AV7, shown in panel A, and AV9, shown in panels B through F. AV7 transduction was sparse and scattered throughout the cortex in cells predominantly observed to be astrocytes. Even sparser labeling was observed in the striatum, thalamus, brainstem, and cerebellum. As shown in the inset in panel A, additional GFP expression was observed in the blood vessels of the hippocampus. So we believe AV7 does not actually cross the blood-brain barrier efficiently. In contrast, for AV9, dense expression of GFP was observed throughout the entire brain, as seen in panel B. Multiplexed IHC for GFP, NUN, and S100B demonstrates AV9 transduction in almost exclusively the astrocytes of the cortex, seen in C. In cerebellum, the GFP expression was observed in Purkinje neurons, as seen in panel D. In the hippocampus, GFP was expression following AV9 administration is present in the astrocytes, CA2 pyramidal neurons, and dentate gyrus granule cells in panel E. And shown in F, AV9 transduction is also present in the choroid plexus in the lateral ventricle. In summary, we believe that AV7 may be a viable option for clinical use, as it is manufacturable on par with AV8 and 9, could be particularly important for heart-specific indications specifically. Of course, further study in non-human primates would be ideal in a complete evaluation of capsid tropism and biodistribution, and studies are ongoing. Overall, AV9 still demonstrates superiority for skeletal muscle indications. Ultimately, we believe that any study that only performs biodistribution via PCR-based method is likely missing key pieces of data. As if we had not performed any imaging studies, we would have concluded that AV9 and AV7 were not meaningfully different, that AV7 was as efficient as AV9 at crossing the blood-brain barrier, and we would have completely missed the transduction of facial structures that aren't sampled in BD studies. So to conclude, I'd like to acknowledge my colleagues at REGENXBIO for supporting this work in many ways, as well as our colleagues at Rutgers University, who we have an SRA with. They perform the cryo-EM, and in vitro, who perform the CFT. And of course, a well-deserved shout out to GenScript, who is our gene synthesis, plasmid construction, and plasmid prep supplier. Thanks for listening.
Thank you, Dr. Yost, for walking us through this study and explores the potential of AAV as gene delivery factors for treating diseases. If there are any questions from the audience, feel free to put them now in the chat box, and then Dr. Yost will answer them for the remaining time permitted. So let's go ahead and get started with some of the questions we already got. First of all, for the glycan array that was mentioned, what were the controls for the experiments, and were there any trends you could see in the AAV7 glycan binding data?
Sure. First, I want to say, again, thanks for inviting me. Thanks for having me at this great symposium. I also want to apologize to the audience. There were 2 videos in there. They were very pretty, spinning, glowing mice videos that did not play. If you want to see those, just shoot me an email or send me a message on LinkedIn. Yeah. So for the glycan array, I think I did mention that we used AV9 as a control and saw that it bound to N-terminal galactose glycans. For AV7, it was a lot more indiscriminate. There were, in the top few hits, there were some fucosylated milk oligosaccharides. There's a lot of variable branching patterns in the glycans, like nothing we could discern as a trend. But for those fucosylated milk oligosaccharides, they have been known previously to be antibacterial, antiviral. So it may be non-specific. We don't really know. The top hit, we haven't been able to have any company synthesize because it's extremely complicated and very expensive. So that's really hindered any further studies on the glycan, unfortunately.
Thank you for the answer. Let's move on to the next one. I think we have maybe time for a couple more. So Could you help explain a little bit more to how the biodistribution was performed?
Sure. We did tail vein injections and then harvested tissues 3 weeks post-dosing. We prefer to chip the tissues to an appropriate size for an extraction, and we typically use Kingfisher. It's a product made by Thermo for our DNA and RNA extractions. We use a rigorous DNase protocol for our RNAs before putting them into the RT reaction. And then in the bad old days, we used qPCR. I think that first BD study was qPCR, and then since then we've graduated to ddPCR, which we much prefer, either using an assay against the poly-A or a transgene-specific assay.
Thank you. Moving on to the next one. So what are some of the limitations of PCR-based biodistribution studies?
I think it's a lot. It's kind of a situation where like it's the best we've got, so that's what we use. But you know, there's a lot of factors in there. You know, how specific is your primer probe assay? Is there a lot of background in the genomes you're looking at, and what can you actually extract viable DNA and RNA from? In this case, you know, we're not going to extract DNA and RNA from odontoblasts. That's just not something that people typically do, particularly for AAV biodistribution studies. But hey, maybe there's a dentist out there that wants to use AAV for gene therapy. You never know. So maybe that's useful information for someone out there. So yeah, I think the limitations are like, where can you realistically look for things using PCR? Where can you actually get nucleic acids from? How sensitive are your assays? How confident are you in, you know, your sensitivity of those assays and that sort of thing?
Okay, thank you. So that's a shout out to our audience. If there are anybody that are interested and they can help, feel free to let us know.
For sure.
Another question that we have here is, what are some of the desirable properties that are less studied and then naturally occur in AAV capsids that may possess? And then why is it important to thoroughly characterize those capsids?
Can you repeat the first part of the question?
So what are some of the desirable properties that less studied naturally occurring AAV capsids may possess?
Yeah, so there's a lot of aspects here. Obviously, I work at a biotech, so we're concerned with how well does a capsid produce. You know, no matter how well it goes to your desired organ, if you can't make it, then it's not useful commercially. So yeah, REGENXBIO owns over 100 of these NAV capsids that have been naturally mined, and a lot of these just haven't been characterized in terms of production. Can you make a potency assay for it? Where does it go in vivo? And is it the same? Does it go to the same places in mice? Does it go to the same places in NHP? Does it go to the same places in human? We just really don't know. So yeah, there's a ton of indications out there where you want, you know, you want to cross the blood-brain barrier or you don't, or you want some heart-specific indications. You want something that maybe goes to the liver a little bit less, because AAV tends to get soaked up by the liver a lot. So that's usually a pretty desirable property. Yeah, I think there's this kind of, to me, it's a balancing act. So, you know, you could have a capsid that produces really, really, really well, but if it doesn't go to any of the organs that you care to treat for an indication, again, useless. So it's like you kind of have to make a priority list of what you think is important for your, you know, potentially clinical use capsid, and decide, you know, how to weight those aspects in the thing you're evaluating.
Got it. Thank you. The next one we have here is, have you tested your AAVs in vitro for blood-brain barrier assays?
Many years ago, we tried to do a transwell blood-brain barrier assay. I think there are much better models now that we have not tried. We did not have great luck back then with those more rudimentary assays. But I think there are much better assays now. If I remember, it's, gosh, I want to say Fujifilm makes one now that's supposed to be pretty good, but we haven't tried it yet in our hands. But yeah, that's definitely something we want to get more into. It's just bandwidth.
Okay, understood. Moving on to the next one. How might the information obtained from the study about AAV7 and AAV9 capsids contribute to the field of gene delivery, and what potential applications or capsid engineering properties could come from these findings?
Yeah, so when we initiated this study, we were really hoping to get a clear answer, particularly for the glycan binding, because that's, kind of a home run of, you get something that binds really well, you potentially do a co-structure, then you make a library in that area, and you can alter the properties, the tropism, and that sort of thing. We still want to do a co-structure with AAVR. There's always engineering opportunities when you have a co-structure. I think AAV7 in particular would, assuming, well, this is a really large assumption, that our findings translate to NHP, which who knows whether it does or not. It would be particularly good for heart indications. If we could further liver detarget AAV7, that would be great. There are some methods of doing that have been described, particularly for AAV9, that may also be applicable for AAV7, depending on sequence identity. But that's not something we've pursued yet.
Okay, thank you. Moving on to the next one. So what are some of the other characteristics that your team is currently finding out about AAV7 to confirm if it's a viable clinical tool?
Yeah, so we've done a lot of studies in NHP since these experiments that involve, actually a bunch of my colleagues presented these at ASGCT. So if you've been to ASGCT in the past couple of years, you've probably seen this. We've been calling it the PAVE 118 library. So we have 118 capsids, a lot of benchmarks in there, a lot of REGENXBIO owned capsids in there, individually barcoded capsids that we put in an NHP, and then evaluate the transduction, the tropism in those animals. Unfortunately, that study, well, our methods now are a lot better than when we performed that study. So the results from that weren't statistically significant, but it did hint at AAV7 still being a very good heart transducer. Whether it's better than AAV9 in that context, that still remains to be seen. Yeah, so beyond what I've presented here for AAV7, that's really the only thing I think we've really touched on as a company. REGENXBIO is still pretty focused on AAV8 and 9. We have some new things going on in capsid engineering, so hopefully soon we'll be seeing something new, but for now 8 and 9 is it. Okay.
Well, looking forward to what the REGENXBIO might have planned for AAV7. So the last question we have here is, what novel findings were uncovered through the CFT, particularly concerning the transduction patterns in facial structures, teeth, and some other tissue? Hang on. Yeah, actually, we have another question come in, but we can answer that after this one.
Sure. Yeah, so we found that there was a lot of transduction in the tongue, which I think people have looked at before with biodistribution studies, particularly for AV9, because it's technically a muscle, right? So other than that, like inside the jawbone, inside the incisors, it's just not stuff that people look at. So yeah, that was a bit of surprise. We really thought actually for the AV9 animal that the brain would light up even stronger, and we were surprised that we didn't see a huge statistical difference between the transduction of AV7 and AV9 in the brain. But I think it's just the resolution is not high enough for CFT to see the kind of differences that we saw in the immunohistochemistry. So like to see that AV7 was only able to transduce those blood vessels very strongly in the brain and not actually cross the BBB was not something that we could have seen from the CFT, which was interesting. So it seems like kind of, you know, every experiment gives you a little more piece of knowledge that you wouldn't have had before, but without everything, you would never have the whole picture.
Yes, those imaging, I do encourage, sending out a message to Dr. Yost to see those videos on her slides. Those are very, very interesting. Moving on to our next question. Were empty capsids used in your studies? And would you expect any biodistribution differences between empty and full capsids?
This is a really good question. All of these vectors were produced in such a way to minimize the amount of empties. That's not to say that they were ever zero, because that's extremely difficult. I wouldn't expect a tropism difference unless there were so many empties that you saturated some of the organs, which would send some other capsids other places that it wouldn't necessarily go. But I think you probably need a much higher, like a very high dose of empties in order to do that. But in this particular study, I don't think I'd be worried about it.
Okay, thank you. So that concludes the session. On behalf of Transcript, we would like to thank Dr. Yost for her time today to answer the questions and then sharing her breakthroughs in gene and cell engineering fields. And our next talk will start at 2:30. In the meantime, you can visit our exhibit halls as well as networking lounges for more activities, and we'll see you back in a little bit. Thank you.
Thank you.